This is a summary of the DNA evidence from the Georgia versus Edrick Faust case, which happened recently. Edrick was convicted of the unaliving of Tara Baker in 2001. I found the DNA evidence really hard to understand, so I wanted to put it into a visual. An SA kit was collected as part of the autopsy back in 2001. Back then, they only screened for certain types of DNA. The first swab was taken from the rectal area, and that was labelled 2B. The second swab was labelled oral, 2I, and the third swab was labelled 2J. All three of these samples tested negative, so there wasn't any more testing done on the samples, and the case went cold. The case was reopened after the passing of the Baker-Coleman Act, where it became possible to open cold cases for reinvestigation. The same three samples were sent to the crime lab. All three samples were found to have male DNA. Each of the samples was then further tested and separated into first and second fraction cell groups. First fraction cells are non-male cells, and second fraction cells are male cells, and that could be any type of male cell. All three of the samples were matched to Tara Baker in the first fraction cells. The second fraction cells of each of the samples had multiple partial DNA profiles. Swab 2B had multiple partial profiles with at least 3 contributors. Because of the complex nature of the mixture and the low-level data, no further testing in the normal way could be done. This is where newer technology, true allele testing, was undertaken. I'll do another video on true allele later. From this sample, there were at least 3 contributors. Tara Baker and Edrick Faust were both ruled out as inconclusive, and the one match that they did find was Chris Melton, Tara Baker's possibly ex-boyfriend at the time of her death. He was one of the alternative suspects that were presented in the trial. Sample 2I was an oral swab. Testing of the second fraction cells revealed at least 2 partial profiles. One was a major contributor and one was a minor contributor. The major partial profile was identified as Edrick Faust, and this was through comparison to the CODIS database. The true allele testing on this sample confirmed the match to Edrick Faust, and Chris Melton and Tara Baker were both excluded. 2J was labelled as oral swab A. More on this in another video. This sample had at least 3 contributors. Again, the data was low level, and because it was a complex mixture, they couldn't do any further testing. After true allele testing, it was shown that there were 3 contributors, and Tara Baker was not included in those 3. Chris Melton and Edrick Faust were both lower level matches. So to summarise, the rectal swab had confirmed DNA from Tara Baker and Chris Melton. Oral swab B had confirmed DNA from Tara Baker and Edrick Faust, and oral swab A had confirmed DNA from all 3. But that leaves still questions about the unknown profiles. It was unclear what type of cells were in the samples, but what we do know is that they weren't blood or sperm cells. Many issues with DNA evidence, in my opinion. Firstly, it was trace DNA, which means it could be from anywhere at any time, and it could have got there in any way. It doesn't give us any information about what kind of cells it was, how it got there, how long it had been there, etc. All of the samples were complex mixtures with low-level data and 2, 3 or more contributors. And the samples were only compared against the 3 people mentioned in the video. In my opinion, the lab practices are highly questionable, and this is because of the mislabeling of evidence being missing from envelopes during the trial, and just generally poor chain of custody. There was actually a fire at the crime scene. When emergency responders arrived, there was a lot of mess, a lot of debris. They had to use water, obviously, to control the fire. One of the most blatantly obvious problems with the DNA evidence in this trial, in my opinion, is the double standards. So, for example, Chris Melton's DNA was explained away, saying it was okay for that to be there, but presence of someone else's DNA was used to indicate that they were guilty, and that is just flawed. I'll be making more videos in the future going over other problems with this whole thing, like the poor investigation that was done back in 2001, and also the possible political motivations behind the whole cold case opening situation. So stay tuned for that. But right now, Edrick really needs our help. He is fighting the system. The system is corrupted and the system is broken, and he really needs any support that he can get. So the QR code links to his a pellet fundraiser. So if you can, please contribute or share, because it really helps.